I hope that you have seen my previous videos on the photosynthesis electron transport chain, the origin of photosynthesis, and RuBisCO. In this video, I will focus on the Calvin cycle of photosynthesis and how sugar is made by plants. The light-dependent reactions of photosynthesis take place in the photosynthesis electron transport chain of the thylakoid membrane. The light-independent reaction of photosynthesis is the Calvin cycle. The Calvin cycle takes place in the stroma of the chloroplast. The Calvin cycle is also called the dark reactions of photosynthesis because it requires no light photons. Do not let the dark name of the reaction fool you. The dark reactions, aka Calvin cycle, occurs during daylight and simultaneously with the light-dependent reactions of the photosynthetic electron transport chain. To repeat, the light-independent Calvin cycle happens in the stroma simultaneously with the light-dependent photosynthetic electron transport chain in the thylakoid membrane. The photosynthetic electron transport chain provides the NADPH and ATP molecules that power the Calvin cycle. The Calvin cycle uses the energy from NADPH and ATP to convert carbon dioxide CO2 and water H2O into the simple sugar, glucose C6H12O6. Glucose molecules are stored in long-chain glycogen molecules that provide medium-term energy storage for all cell functions. The glucose molecule can provide the basic building block for all other sugars, as well as fats, proteins, and nucleic acids. So, glucose production in the Calvin cycle is key for the survival of all aerobic life on Earth. In a previous video, I went on a tangent to discuss the controversies surrounding RuBisCO, the most important enzyme in the Calvin cycle of photosynthesis. As we left off our discussion of photosynthesis, the photosynthetic electron transport chain of the thylakoid membrane has made NADPH and ATP for the chloroplast stroma. The NADPH and ATP are the energy sources the Calvin cycle needs to make glucose from water and carbon dioxide. 3 Calvin cycles run in parallel. The Calvin cycle begins with the RuBisCO enzyme adding a carbon dioxide molecule to a 5-carbon molecule, called ribulose 1,5-bisphosphate, C5H12O5, PO3, RuBP for short. The RuBP is present from the previous cycle. The RuBisCO enzyme splits the 6-carbon intermediate into 2 3-carbon chain molecules called 3-phosphoglycerate, 3-PGA. Remember, this step repeats 3 times, so we end up with 6 times 3-PGA. In the graphic, the 2,3-phosphoglycerate is reduced and energized by accepting a phosphate from 2 waiting ATP to become 2,1,3-biphosphoglycerate molecules, abbreviated as 1,3-BPG. These paired reactions occur simultaneously in the 3 parallel Calvin cycles. The ATP was produced in the photosynthetic electron transport chain. Upon losing 1 phosphate, the ATP becomes ADP. The ADP leaves the Calvin cycle to wait for the ATP synthase of the photosynthetic electron transport chain to recharge it with another phosphate. So, for the 3 Calvin cycles, the equations are 3CO, 2 plus 3 RuBP, reacts into 6,3-PG. 6,3-PG plus 6 ATP reacts into 6,1,3-BPG plus 6 ADP. Upon the formation of the 2,1,3-biphosphoglycerate 1,3-BPG, the 2 NADPH formed in the photosynthetic electron transport chain adds more energy to the 2,1,3-biphosphoglycerate 1,3-BPG by adding an electron to it. The electron knocks off 1 phosphate pi group off each 1,3-BPG, and the result, 2-glyceraldehyde 3-biphosphate, G3P for short. These paired reactions occur simultaneously in the 3 parallel Calvin cycles. The NADPH becomes the oxidized NADP+ and returns to the light-dependent chain for a recharge. 6,1,3-BPG plus 6 NADPH reacts into 5G3P plus 1G3P, plus 6-phosphate plus 6-NADP plus. The G3P becomes the basis for reshuffling of the carbon atoms. 5G3P remain in the 3 parallel Calvin cycles. 1G3P. C3H7O3PO3 goes off to become half of a glucose molecule, C6H12O6. A second turn of 3 parallel Calvin cycles would result in a second G3P. In an ideal world, the conversion of 2 G3Ps to glucose would simply add the 2 G3Ps to 2 waters, resulting in glucose or fructose and 2 molecules of phosphoric acid H3PO4. Biochemists have found the real-world version of converting G3P into glucose. Involves tracing G3P through an enzymic conversion by triose phosphate isomerase to become the 3-carbon dihydroxyacetone phosphate DHAP. The DHAPC3H7O3PO3 is bonded with a second G3P to get a 6-carbon ring molecule, fructose 6-phosphate, C6H13O6PO3. The phosphate and spare hydrogen is lost to the solution to leave fructose or glucose, both of which have the chemical formula C6H12O6. The DHAP plus G3P reaction is guided by the enzymes aldolase and fructose 1,6-bisphosphatase. In full, 6CO2 plus 18 ATP plus 12 NADPH plus 6H2O results in C6H12O6, aka glucose or fructose. To complete the Calvin cycle, the 5,3-carbon G3P are acted on by enzymes and 3 ATP to regenerate 3,5-carbon RuBP, ready to restart the 3 parallel Calvin cycles. Note: 5 3-carbon G3P have a total of 15 carbon atoms, and 3 5-carbon RuBP have 15 carbon atoms. It just takes enzymes and the energy from 3 ATP molecules to cause the transformation reactions of 5G3P into 3 RuBP. The real-world regeneration of 5G3Ps into 3 RuBPs is very messy and involves steps through fructose 6-phosphate F6P, erythrose 4-phosphate E4P, xylulose 5-phosphate XU5P, cetoheptulose 1,7-bisphosphate 7C, cetoheptulose 7-phosphate S7C, ribose 5-phosphate 5P, ribulose 5-phosphate RU5P, and then finally ribulose 1,5-bisphosphate RuBP. The regeneration steps follow. In step 1, we already made fructose 6-phosphate F6P by adding DHAP to G3P. For step 2, the enzyme transketolase TK removes 2 carbons from fructose 6-phosphate F6P to make erythrose 4-phosphate E4P. For step 3, the transketolase TK adds its 2 carbons to another G3P to form xylulose 5-phosphate XU5P. For step 4, a phosphopentose epimerase enzyme acts to convert the XU5P into ribulose 5-phosphate RU5P. For step 5, the aldolase enzyme acts to join E4P and a DHAP to form cetoheptulose 1,7-bisphosphate 7C. For step 6, the enzyme cetoheptulose 1,7-bisphosphatase cuts the 1-phosphate from the cetoheptulose 1,7-bisphosphate 7C to leave cetoheptulose 7-phosphate S7P. For step 7, the enzyme transketolase TK removes 2 carbons from the cetoheptulose 7-phosphate S7P to leave ribose 5-phosphate R5P. For step 8, step 3 repeats, and the transketolase TK adds its 2 carbons to another G3P to form xylulose 5-phosphate XU5P. For step 9, a phosphopentose isomerase enzyme acts on R5P to convert it into ribulose 5-phosphate RU5P. For step 10, in a repeat of step 4, a phosphopentose epimerase enzyme acts to convert the XU5P from step 8 into ribulose 5-phosphate RU5P. There are now 3 RU5Ps available. For step 11, the enzyme phosphoribulokinase adds a phosphate from ATP to the 3 RU5Ps made in steps 4, 9, and 10 to make 3-ribulose 1,5-bisphosphate RuBP. Step 11 repeats 3 times to end up with the 3 RuBP needed for the next Calvin cycle. In the next set of figures, I show 3 researcher presentations of the Calvin cycle of photosynthesis. So that concludes my discussion on photosynthesis. In summary, I covered the light-dependent reactions of the photosynthetic electron transport chain. I speculated on the origin of photosynthesis. I covered the light-independent reactions of the Calvin cycle. I showed an ATP and NADPH that are produced in the photosynthetic electron chain are used to power the light-independent Calvin cycle. I devoted a whole separate discussion on the RuBisCO controversy and whether the RuBisCO enzyme does a good job or bad job compared to other organic enzymes. I hope I showed that RuBisCO does a fair job of converting inorganic carbon CO2 into organic carbon molecules that can be used in biology. Maybe you have a different opinion of RuBisCO. I briefly discussed how the chloroplasts in every eukaryotic plant cell might have been independent cyanobacteria that were engulfed and formed an endosymbiotic relationship with the host eukaryote cell maybe 1 billion years ago. I hope that I have given you a vision of the complexity of photosynthesis and given you some respect for the photosynthetic organisms that produce the sugars that form the base of the food chain, on which 99.9% of all living organisms depend. Without chlorophyll's ability to convert sunlight into sugar, life on Earth would consist of only a handful of extremophile microbes that can digest rock and gases from deep ocean volcanic vents and make their energy via chemosynthesis. Without photosynthesis, we would have no multicellular organisms or the diversity of life forms we see on Earth. Humans and civilization would never have come into being without photosynthesis.
Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.